So, very good morning, everyone, and welcome you all for the January guest lectures. And today we have two lectures. The first lecture is by Associate Professor Tarja Sironen from University of Helsinki. Let me get a minute to talk about her. So Tarja Sironen is an Associate Professor of Emerging Infectious Diseases at the University of Helsinki, Faculty of Medicine and Veterinary Sciences. She is a virologist leading a research group studying zoonotic microbes focusing on rodent and bat-borne microbes and their drivers in their emergence. She's also the director of Helsinki One Health Network, and she leads the project Resilience and Just Systems, combining life and social sciences across 8 faculties of University of Helsinki to search for sustainable and just solutions to global challenges, including pandemics. So today she's going to talk about a One Health approach to pandemic preparedness. So over to you.
Okay, good morning, everyone, and thank you for having me here, and thank you for the kind introduction. So what I thought I would do today, I have picked up 3 examples from our research that we use to find solutions to hopefully one day prevent epidemics or even pandemics. So I will talk about 3 viruses. I will talk about coronaviruses, of course, the avian flu outbreak, and then my favorite topic, which is Bombali Ebola virus in the end. And I will discuss the methods and approaches that we use. So indeed, from my background, I'm actually a biologist from University of Jyväskylä. But already in my master's, I got interested in viruses, how they interact with their hosts, and then moved on to University of Helsinki a long time ago, actually, already. I indeed have the honor to be the current director of Helsinki One Health Network. So this is a network initiated by Professor Olli Peltoniemi. And once he was appointed as a dean, I took the job to be a director for Helsinki One Health. My vice director is Anna-Mari Heikinheimo, who is studying AMR, zoonotic AMR, and Topi Tanskanen is our coordinator. And we embrace this, I think, very wide One Health definition that has been rewritten now a bit more than a year ago. And I like this because it takes action and it takes a positive viewpoint. So to me, One Health is improving health, whether it's improving human health, animal health, environmental health. So I don't think there can be human health if there is no animal health or environmental health. This definition also talks very much about collaboration and communication and capacity building. So we can't solve these global challenges, One Health challenges, if we don't work together. So I am a virologist. I'm really interested in the viruses and how they interact with their host, but that's not going to solve pandemics alone. So we need to work together across different disciplines and sciences if we really want to solve these global challenges. And I do think One Health approach will be essential in that. Because of this understanding that we need to work together across disciplines, we built this project that has now started last year, which was already mentioned, so Resilient and Just Systems. And we truly aim to develop interdisciplinary research, bridging life and social sciences. And it was quite a long process to build this and even learn how to talk across the different disciplines and sciences. But now that we have finding the common language, we are getting very excited of all the opportunities that we can do and what we have. For us in University of Helsinki, we found these 4 themes that we want to focus on and what we want to build further. So I'm obviously mostly in the theme 1, infectious diseases in changing environments. And this can be anything. This can be understanding how wildlife is hosting or being a reservoir for these pathogens, or how can we develop drugs or vaccines, or it can be really looking at the structures of the pathogens. But then we move on to theme 2, which is the health of the animals, wildlife or domestic. And that, of course, from my perspective, and it's just the infectious disease perspective, it has an impact on the infectious disease outbreaks. So an animal that is not healthy, that is under stress for whatever environmental reasons, climate change, heat, or something else, some pollution in the environment, it is also more susceptible to diseases. It has a different immune response, and it will have an impact on the infectious diseases. Or we can change the land use, we can restore areas, and we can change the biodiversity, and it will have an impact. Then on the theme 3, we talk about sustainable food and water systems and do research on that from a very broad perspective. If I take the hat of the infectious diseases, I'm thinking of production animals, how we farm them, and how that is actually leading to increased circulation of certain pathogens, for example. And then finally, we move on to the society. So I can deliver whatever results. I can tell that I will show you that SARS-CoV-2 is airborne and you should wear a mask. But if I do that in my lab, it doesn't mean anything. I have to translate the results. I have to talk to decision makers, and I need to talk to people and talk about the results and all the way to the society. And I have to talk why vaccines are useful. So there's a lot of hesitation to take vaccines, for example. And me as a scientist, for me, it's clear that vaccines work, but that's not the whole truth. And so how do I communicate this to people so that they actually take the vaccines? There I need the social scientists to help me to understand how to do this. And all of this we're trying to bring together. But this is what my research team does, and I will focus on this part here. So I'm interested in cross-species transmission. So when viruses jump from one animal to another or to people, what happens to the virus? How does it evolve? And what are all the different factors driving this cross-species transmission? Whether it's viruses that are my favorite topic, and we also do a bit of work on antimicrobial resistance, but I will talk about viruses today. So I have studied initially rodents. Since my PhD thesis, I've been interested in rodent-borne diseases. I tried to switch on to something else, but now I'm back with urban rats, for example. And then about 10 years ago, I started to work with bats and bat-borne diseases. I do a bit of work on ticks as well. I will not talk about those today, but there is a lot of work on how ticks are now spreading in Finland due to climate change, spreading new diseases, both to companion animals, to domestic animals, and to people. So there's a lot of research, and if you're interested in that, maybe we can discuss that later. We also study mosquito-borne diseases in our unit. But we study the reservoirs, the bats and rodents in the wildlife. We look at spillover to domestic animals. I work a lot in Africa, but I also work in Finland, and this last year mostly in Finland actually. And then we look at spillover to people as well, and work in mainly with BSL-3 level pathogens in Helsinki, and I also try to communicate the results. So when we talk about disease emergence, I recently went back to this slide that I had already some years ago. So this is from April 2018. And that was a time when there was a lot of big projects on virus discovery. And Bill Gates gave an interview with this quite alarming title saying that there is a disease coming that could kill 30 million people within 6 months. And this was 2018. And there was a lot of discussion. How can we prepare? What should we do? And the challenge is here. So it has been estimated that there are 87 million different viruses on the globe. We know about a bit less than 5,000 of those. So which one of those 87 million viruses is going to be the next pandemic? We can't study all of these viruses one by one, what kind of biological properties they have. It is just impossible. So how can we narrow down what we study? This is again something from 2018, so time before, just before COVID. So this was a watch list by WHO. And you can see the list. Diseases threatening a public health emergency. All of these are viruses. All of these are zoonotic, so coming from wildlife to people. And all of them have an RNA genome. So there's already some narrowing down. And you can see coronaviruses there, known coronaviruses. So they were kind of on the watch list. So we were supposed to be prepared. There is a disease X, noting that we can't really know what is going to be the next disease emergency. And this is now public health and talking about human infections. So what about all the animal infections? They have a huge impact. If we have an outbreak, an animal disease, it has a huge indirect impact on us people as well. But here, of course, the focus is just on us humans. So 1 year later, this one came, and I'm somehow very, the spikes on the virus surface have really burned in my mind. So COVID-19, so SARS coronavirus 2 started to spread at the end of 2019. And funnily enough, I was appointed to my position in January 2020 on emerging infectious diseases. And I had plans, but I had to ditch them and focus on this pathogen. And I will not go too much into detail. It's been 4 years, a bit more than 4 years. We have more COVID-19 cases than ever in many areas. So the virus is still there. But are we really caring about that? Should we care more? Should we wear the masks? Should we stay home and have Zoom meetings again? I don't know, but that's maybe something to discuss. Why do we accept all these infections at the moment? So what we did in Helsinki, we teamed up with pretty much everyone else at our university. All the other research was pretty much stopped. So all the virologists worked on coronavirus and many others as well. So this was a very, very interesting time. So many people got interested and wanted to work on this virus. But these are the things we focused on. So we, for example, produced a vaccine for animals that was actually in use in Finland, emergency use. We have been also working a lot on the drugs and drug development, on the immune responses. And I have been particularly interested in the modes of transmission of these viruses. And we use many animal models to do this. I am also more interested in the animal side. So these are actually very, very common viruses in animals. So there is hundreds or even thousands of coronaviruses in many, many animal species. And I think there is a lot to learn. And we have been screening rodents and bats in Europe, for example, in Africa, in Europe. And we find SARS-CoV-2-like viruses also in European bats. Should we prepare for them? Will they cause the next outbreak? That is what we try to estimate now. We can't do that based on the sequences. So we need to study the viruses still. But I wanted to bring this one up since we talk about interdisciplinary research. So in March 2020, Ville Vuorinen, he's a physicist, aerosol physicist, so interested in the particles, how they move in the air in indoor spaces or outdoor spaces. And he actually called me and asked me if this virus is airborne. And I was like, no, it can't be. I mean, it's a droplet, so it can't be. It's too scary if it's airborne. And we ended up discussing 2 hours on all these aerosol dynamics and started this project, a fantastic project that still continues. And by modeling and by actual experimentation, we and many others show that this virus is actually airborne, and that's one of the reasons why it was spreading so efficiently. So combining really hardcore physics to virology was quite a journey. And what we did in our lab is we studied the viruses using aerosolized viruses. We used animal models, cell culture models, and we went on to people's homes to collect air samples. We went to hospitals, we went to mink farms, and we showed that we can find this virus in the air. It's still not very efficient. It's not sensitive like the wastewater surveillance, but maybe in the future this is another way of doing surveillance. And all of this led into this project, E3, Excellence in Pandemic Response and Enterprise Solutions. So we combined 8 research organizations, 22 companies, and we have been looking for solutions for healthy air, indoor air, whether it's offices, whether it's hospitals, whether it's well, basically anywhere. We are looking at daycare centers, for example, at the moment. So quite an interesting journey, and you can really work together to deliver the solutions. So I'm still coming from the hardcore sciences, but working directly with the companies. And this was new to me and very exciting. And we have, for example, in the daycare centers, we purify the air with these new solutions that we provide, and we show that actually there's 30% less respiratory infections in the daycare center because of this air purification, which of course is a huge difference if you have small children and at the daycare, and you think of all the days you are sick or your children are sick. So if you cut down 30%, that is a huge impact. Of course, we don't know exactly what are the viruses and why that is, but anyway. And this is a very recent paper, not ours, but coming from Asia. So now I'm moving towards avian flu. And so this aerosol surveillance is, I think there is a lot of effort to develop this to be something efficient. And it is used in live animal markets where they have chicken or any birds. And in this study, you can see that they show that when it's the avian flu season for the birds, 100% of the air sampler give a positive result. You can find the virus in the air. So you don't have to go and collect the animals or test the animals one by one. It is enough to collect the air sample. And the sampler is so it's the worker that has the sampler. And you can see that you can find by PCR in 100%, and in 50%, it is infectious virus that you can see. But then again, if you are out of the season, so it's not the avian flu season, you can't find the virus. So could we use this air surveillance like we use the wastewater surveillance at the moment, maybe in the future? That's something we also try to work out. So then I come to avian flu and last year. So I do have to say that before last summer, I wasn't really doing research on avian flu or influenza A. So it was my colleagues who were doing, but I have been working on the farms and looking at the other infectious diseases of fur animals. So I was in contact with the veterinarians, with the farmers, and they started to contact us that there's something new, something happening that they cannot handle. So we went to the farms to see what's going on. So before the farms, there was a lot of reports in Finland on the bird mass die-outs. So there were hundreds or even thousands of birds dying, and it was H5N1. And when we went to farms, our authorities had already confirmed that it's H5N1 that we see in the fur animals. So we knew what we were doing research on. But what was happening in the foxes and in the mink was something completely new. So we thought that we really want to see how is it causing disease in the animals? What is the pathology? How is it transmitted? Is it transmitted from animal to animal? And that's actually still something we are working on. And why this is scary, of course, is influenza A is the most likely cause of a new pandemic. And so if you want to bet your money on the next pandemic, I would bet on influenza A, simply because it has done so for many, many times before. And it is very widely spread in the animal hosts. And H5N1 has been known since 1996. So it was first found in birds, and then it immediately, a year later, it was found in people, and we know it's causing severe infections, or back then at least we knew it was causing severe infections for people. We don't actually know at the moment what is the pathology in people, because we don't really see human cases at the moment so much. But what we do know is that if you passage this virus coming from the birds in mammals, ferret is the model, it changes how it is transmitted. So you can actually see it's coming airborne. And we know what the mutations are. We know we need 5 mutations that this virus is efficiently transmitted between mammals. So we can actually follow these mutations and we can study these. So influenza A is very well-known virus throughout the years, unlike SARS-CoV-2 was. So this is the starting point, and also luckily we were part of this durable project, which is a research network across European research institutes, and they had the resources available that we can actually go on the farms and do this research. And here you can see a PhD student of mine, Rasmus, collecting air samples on the farm. So this is a mink farm, and what we went to do is to sample the animals, to look at the disease, what kind of symptoms, signs of disease there are, and then collect samples and do pathology on the animals. And this is what we did for 2-3 months at the end of summer, early fall, when the situation was going around. So looking back, our authorities have now surveyed all the farms, and there's about 60 farms out of the about 400 farms that were affected. So this has really spread widely in Finland, and we went to do a thorough analysis on 5 of the farms, and we are working on the manuscript, but this is now so far unpublished data. So if you think of influenza A, you think of respiratory symptoms, and that's what we saw in the foxes. But we also saw a lot of neurological symptoms. So in the mink, there was hardly anything else except the neurological symptoms. So there were changes in behavior and a poor growth of the animals. But in the foxes, we had very, very clear respiratory severe symptoms. What is challenging is that some animals seem to be completely asymptomatic, or you don't really have strong enough symptoms to notice them on a farm of thousands of animals. But then we also had fatal infections, very, very severe infections. I don't think I have ever seen myself so severe infections as we look at the pathology. We look at the antibodies together with Martin Beer and his group, and you can see a huge difference. So on certain farms, we have a small outbreak. All the affected animals seem to die very fast, and the virus doesn't spread, and we don't see antibodies. On another farm, 80% of the animals, almost 80%, have antibodies. So the virus has spread, unnoticed, infecting the animals, they get immune. We don't yet know why that is. So it's very difficult to predict what will happen next in the animals. What we did saw is a very high viral load. So you can find the virus anywhere in the secretions. You can isolate the virus, you can grow it in cell culture. So it's really easily, so there's a huge infection pressure. So no wonder it was spreading so widely. So far, no human cases. So none of the farmers, none of the veterinarians, none of the animal caretakers were infected. Does it mean that we don't need to worry about this virus? Well, I don't think so, because this is an extremely severe infection for the farm animals, for the birds, for the sea mammals that die. And this virus is circulating all across the globe. So I do think we need to pay attention, even if it never causes a human infection. So it is a severe animal health and welfare issue in any case. We looked at the sequences, and of course our authorities have done most of this, but we added quite some sequences on our own. And you can see down, all you need to see is the colored ones. Don't worry about the details. So down there in green, you can find the bird-borne sequences, and in red, you can see the mammals, and you can see a mix. So it did originate from the birds, jumped onto mammals, and started to spread there. Up there, you see a farm 4. So this is something we found late in the fall, so something completely different, and we're still working on. It's a very different virus, killing the animals very quickly, and we are trying to understand why that is. But it was circulating somewhere that we didn't really know. And here, another view, again, the colors are the important. So the ones from birds are in green. You can see in red the ones from mink, and the foxes in blue. So we mostly found this in blue. And you can see this time analysis showing that this virus arrived to Finland in March. What happens in March? That's when the migratory birds come to Finland, and that's when it came from somewhere, wherever. And then 2-3 months later, it jumped onto the mammals. And you can see it by analyzing this sequence data. And this is the thing about influenza A, that we really know this virus very well. So we can do this kind of analysis. And then we see a mess. So a lot of introductions from the birds to the farms, and then from one farm to another. So a very wide, effective transmission on the farms. So why is it now in the mammals? This virus has been circulating for 30 years. So why is it now jumping? And it's not the fur animals only. It's sea mammals, it's wild foxes. And one of the things is here, so which is climate change. So there is a lot of papers showing that climate change is impacting the migratory bird flyways. Some birds are more affected than the others. And those that are affected change their routes. They come into contact with new bird species, and the virus finds a way to jump to new birds. And there's more evolution of the virus and more new infections in new species. And of course, this is a disease of poultry. And there is a huge increase in poultry production across the globe at the moment. It is bound to have an impact on all avian influenza viruses and increase their circulation. So now we are waiting for the migratory birds to return, and let's see what kind of virus they bring, and are we more prepared on the biosecurity of the farms. But then finally, I'll move on to Kenya and the research that I do in Kenya. Why we work there is that many of the factors, environmental drivers of disease emergence are very clear and strong there. So these are developing countries or areas where there's a lot of land use change. There's a lot of change in the practices of food production, for example. The climate change is very pronounced in this area. This is a biodiversity-rich area, but because of the changes, there are also changes in the biodiversity. So this is an area where we can study these phenomena very well. And we work at the Taita Hills area, where we have a research station. That's our new building that was just built last year. And this is particularly interesting area because we also have the elevation, so we can go high up on the mountains, or we can go into the lowlands, and it's a very, very different environment. So you can, in a small scale or small area, you can study all of these different environments. There's also some restoration efforts, so we can also study how that impacts the disease dynamics. And on the left, you can see the core team. So on the left is Dr. Paul Webala. So he is a bat conservationist and an ecologist, without whom we wouldn't do any of the bat work. Then it's my colleague Esti Korhonen from University of Helsinki. She's an expert on the mosquito-borne diseases. So she's studying dengue, chikungunya, onyonyong, all these viruses that you can find in the mosquitoes. Then we have Dr. Moses Masika. So he's a doctor, infectious disease specialist, and really looking at the burden of disease for people. And then it's me, and then it's my PhD student, Josef Ogola, who is a veterinarian and looking at then the veterinary side of these diseases. So we need people from different disciplines to do this work. We also have geographers in the team, and we also have social scientists in the team. And this is what we want to do. So we study the bats. We focus on bats that like to come to human buildings, so that are really in close connection to people, and there is a possibility of spillover. We look for the viruses, we do surveillance, we look at spillover, whether it's the domestic animals or whether it's the people. So we try to look at what viruses are there spilling over. Is there something, silent circulation of pathogens that we don't see? And in this area, there is very little known. So if you have a febrile patient, you usually think of malaria, you think of TB, or you have a patient that has gastrointestinal infection. So then you don't really they don't really know what are the viruses causing the disease. So we are looking into that, really just basic mapping. What are the causes of disease? And if we come to the veterinary side, well, there is Rift Valley fever. There's rabies. There's some things that are known, but nothing is really tested. So there is a lot to do for us just to kind of map what are the risks. We improve diagnostics locally so that we could do this on site with multiple methods. Really simple point of care testing all the way to NGS on site. We do a lot of modeling, so we collect environmental data, data on the diseases, remote sensing geographical data. We try to build it, put it all together, and really see what is driving disease emergence. And we are now also adding in some work on social media, so picking up signs and discussions that there's something happening. There's diseased animals or diseased people in a certain area, and we try to confirm, is it truly an infection? That's something we want to do in the future. So we've been to Kenya since 2015. And the message here is that it takes a long time to really build the collaboration. And we really hope to continue this, and we have the resources to do that now. And as you can see, we focus on mosquitoes and bats, but we also do a bit of work on rodents and ticks as well, and the domestic animals and the human samples. We work in 3 areas. In Busia, close to Lake Victoria, it's a highly biodiverse area. We work in Nairobi, in the slums of Nairobi. We have been collecting, for example, rats, which are plenty in that area. And then we work at the Taita Hills area, and then also in Mombasa. So very different environments that we look for. And this has been our main topic, since this is the first virus we actually discovered there in the bats. So we found an Ebola virus back in 2018. So in Kenya, Ebola viruses have not been found before. So there was the thinking that there's no Ebola there. But in our initial study, we wanted to sample all kinds of bats just to see what's in there. And we know there is Marburg, so we thought that we start with the filoviruses, and immediately we found this Bombali Ebola virus, and I'll show you soon how we did it. So it became the most important study subject for our research. We don't have a BSL-4, and we don't have an isolate. No one has an isolate. So there is no Ebola virus isolate from the bats. That's my dream. So maybe that will happen one day, not in our lab, but by our collaborators. But you can do a clone, and there is a research group in Germany that has shown that this virus can infect human cells. So it is potentially a pathogen, but of lower disease severity. So an interesting model virus. There are no human cases, although there are antibodies found in patients. So does it cause disease or not? We're still working on that. It can be found in 2 bat species, insectivorous bat species that like to live with people in houses. So there is a high risk of infection, or at least there is a human-wildlife conflict. And in this area, you have all the domestic animals as well. So are they susceptible to this Ebola virus? We, for example, know that swines are pigs are having antibodies against Ebola viruses. So it is possible. It's something we would like to do in the future. If you look at the virus, it is closely related to those causing disease. So that's something we try to figure out. Like I said earlier, based on sequences, we can't tell if it's causing disease or not. This Ebola virus is very fast becoming the most reported one from the bats. So it's reported already in Guinea, in Sierra Leone, in Mozambique, in Kenya, and I also know it's coming from Tanzania as well. So it is widely spread, and seems to be quite common in Mops condylurus bats. And how we did. So how to detect a novel virus, how to do virus hunting. So we initially collected all kinds of bat species that we could, and we screened them with this pan-filovirus assay that we had developed in an EU project called Ebola Mondrad. And so this is a PCR that should detect any known or unknown filovirus. So now we got to test it, and it worked. So we found one that seemed to be positive. We put all of the RNA to Illumina MySeq, and we actually got a full genome of the virus by metagenomic analysis. And we found the virus there. And we have our own protocol on the virus discovery, because this is actually quite challenging, how to detect the unknown. So, and we are working on a new version freely available and easily usable version of this lazy pub, but it's already freely available to anyone who is interested in virus hunting and detecting novel viruses. And now we move on to really with a specific PCR, which is far more sensitive, looking for further positives. Immediately as we found this, we went back to Kenya and we set all of these tests that are now available at the Gavi Medical Research Institute. Something that is interesting, I talk about the transmissions, how are viruses transmitted from one animal to another. We actually found Ebola in the lungs of these bats. So if you think of Ebola, you think of hemorrhagic infections and bloodborne infections, but this one is actually in the respiratory secretions. So could this be at least in the droplets or even airborne? That's something we are looking into in our work. We also really want to look at the pathology. So this is done at the Faculty of Veterinary Medicine. So we collected some of the whole bats, gave them to our pathologists to have a look at the bats, and they showed that there was really, these were healthy bats. I mean, they have all kinds of parasites and all kinds of infections, but they are the same on the Bombali positive and the Bombali negative. So the Ebola virus doesn't cause a disease for the bats. So it seems like it's the respiratory host. And this is something very unique and not really done before. So I have the highest respect for our veterinary pathologists who can look at whatever species I bring to them. And then I want to mention my colleague Chris Forbes, who is doing the bat work, and he is focusing on, so he's a disease ecologist. So he wants to understand the ecological perspective of Ebola virus circulation. And he is doing this longitudinal monitoring, so tagging the bats. So they have these small tags on their wings, and we recapture them. And as you can see, we can capture them more than a year later, the same bats. So we try to follow the diseases. We try to follow where these bats go, how they change from one house to another, what kind of houses do they like, and can we somehow find solutions and suggest how to build houses where the bats don't actually come in. And they also have a lot of studies on the connections of not only the people but their animals with the bats, and there is a lot of possibilities for transmission. This is Dr. Riley Jackson, who is in Chris's team, and she also, while doing this longitudinal monitoring, she also did a bat education program in the rural Kenyan communities. So we also try to educate people on bats, on the good things about, on the cool facts about bats, how important they are in the ecosystem, and how to also avoid the diseases, while also preserving and conserving the bats. So this is also something we really would like to continue. I think it's very exciting also to do as part of the work and part of the solution also. So this is what I had in mind. I'll quickly show the acknowledgements, but I will only show it quickly because I'm sure I'm not having everyone there. Concerning the work in Kenya, I really want to acknowledge Essi Korhonen and Chris Forbes. And then the work with avian flu is really done within the Durable project, and Martin Beer has been the most valuable colleague in this project. And I thank you for your attention.
Thank you very much. It was very educational and very nice to hear all the detailed aspects of the book. But as a center of one university of Helsinki, how do you justify the work that you do, for example, in the How do you make the general public, also the fathers, the others, to appreciate what you do and to agree with the efforts that you're putting?
That's a very good question. So we want to address global challenges. So we in Northern Europe are not separate from the world. And if we think of infectious diseases and people traveling within a day across the globe, or transporting animals from one continent to another. So we have to work on these global problems, and we can't do that if we only stay in our small bubble in Finland. So we have to do this elsewhere. And for me, the work in Kenya, it's just the perfect setting to do this, where these phenomena are very clear and changing. How do we communicate about this? That's a good question. I think science communication is not easy. It's not something that comes by nature for many of the scientists. We like to stay in our own lab and just work. So all I can say, just go out there and start to talk about your results. It actually helps you to clarify your thinking. Why are you doing this? And once you do that, and once you go and ask or respond to all the questions, that's how they get convinced. So you just have to go and talk and communicate and learn how to do that. It's a completely different thing to talk to a general public compared to your colleagues in a university lecture. Yeah, so it is coming from wild birds, and at least at the current way they build the farms, there's access. They are really open. They are these open barns or sheds or houses. So the birds are there, and there's of course a lot of food available for the birds. So you can actually see that when the food truck is coming for the animals, also the birds arrive immediately. And once you go in the farm, you can really see that the birds everywhere and the dying birds if they are diseased. So it is coming from wild birds and a contact. It is of course a possibility that it comes through chicken, but then if you think of poultry farming, the biosecurity there is on a completely different level. But of course, that is, if there is something happening, then that's another possibility. So the wild foxes and the wild sea mammals, it is thought that they eat the dying birds in the wild, and by eating they get the infection. Yes, so far, yes. Yeah, you have to prevent the birds or whatever pest animals actually. So you just have to build better ways to prevent them from coming to the farms. It sounds simple when I say it, but it's not. Yeah.
We spend thousands of millions of the dollars or the euros in these projects, and then we generate quite a lot of scientific data. But when we translate this scientific data, turn the information to the public, what are the challenges we have faced, especially in communication?
Well, I think there's two challenges. So first of all, us scientists, when I talk about the results, I like to be very vague. So based on this test system, it looks like this, but under another test system, we tend to be too vague. So that's unprecise, because that's how we used when we write a scientific paper. You have to look at your results from different perspectives and look at your, what is the next thing to be done, and you can't do that when you have to simplify your message and find the core message when you communicate that. So that's one thing. The other thing, of course, is that the results are not always the ones that you kind of hope to see, or the general public hopes to see, or there is an partner who doesn't really want to see the results. So it's not an easy. It might cause a need to invest in biosecurity or whatever. So it's not always the hoped message. So how do you deliver that? Still, this is based on our research. This is how it looks like, and we have to deal with the result, although we don't like the result. So that's something I find challenging, and you really have to believe in your data, and you have to do good research, and hopefully someone even repeats your results so that you have more and more data, and you have to firmly stand behind your results, and that's what I find challenging sometimes.
So,
Or I can repeat your.
Can you understand what I mean?
Yeah, so there is a question on the collaboration with the private partners and industries, who of course have their own interest. Well, the key thing is to make the contract and the agreement before you start the research, that no matter what the result is, even if it's against the company's interest, you are allowed to publish that or discuss. So to me, that's really the key thing, and to make sure that my industry partners know that I will publish the results even if they don't like them, and you have to put it on paper. But to me, this project, that has been very easy actually to do. So no one has been really arguing against that, but you just have to do the paperwork. And then of course we divide our tasks. So we actually, in this project, my work package is really looking at the really kind of core virology. So we study the virus in the lab and how long it lasts in the air, and when does it start to die out and things like that. And we try to develop the diagnostic methods more like. So then we deliver our data. So that's part of the deal, so that they kind of get the data even before we publish it, the companies. So that's their actual benefit from this project. So and then of course we discuss that what kind of information they would need. So is that something we can consider while we do research? But it takes a lot of discussion, and you have to be very, very transparent about that. And all of our agreements and basic ideas are freely available online, so you can see how we built this project. So this has been new to me, but there is a solution on how to do it. You just have to put the extra effort into it.